several mechanisms for hormonal control, and one of them is called the fixed membrane receptor mechanism, and this always deals with water-soluble hormones. So that would be, amines, proteins, glycoproteins, so on and so forth. They could all conceivably use the fixed membrane receptor mechanism. And then they're going to have a mobile receptor mechanism that will deal with steroidal hormones. Okay, so that could be, estrogens, aldosterone, cortisol, what are some of the others? Testosterone, so on and so forth. All of those are steroids. Now, if the way this works is, here I've got this chemical messenger, this steroid, and the goal here is that the hormone needs to come in contact with a target tissue or a target cell. And of course, when a target cell is going to be doing some sort of work, the apparatus for dealing with that is going to occur inside the cell. So what happens, since it's water soluble, no problem transporting because it will dissolve in the universal solvent called water. And so it's in the plasma, it gets into the interstitial fluids, and now it comes in contact with this particular cell. But I've got a problem. What I have here is I've got a phospholipid bilayer, and you guys remember this is hydrophilic, hydrophobic, hydrophilic. Hydrophilic, hydrophobic tails, those fatty acid tails, hydrophilic. And the issue here is my water-soluble hormone will not dissolve in those lipid tails. So it cannot enter into the cell to have some sort of an effect. So the way this will work is that it's going to have a particular receptor on the cell surface. And the way this particular one works is it is going to take, once the hormone attaches to the receptor, that will enzymatically convert an ATP molecule into an AMP molecule. Okay? And it has a cyclical form. They'll call it cyclic AMP. Now, in this case, we're going to call this cyclic AMP as the secondary messenger. Okay? And there are a lot of these water-soluble hormones. And the secondary messenger is always cyclic AMP. It's always cyclic AMP. And so what happens, now there's going to be all sorts of unique hormones. You can have some that are, we could just sort of, there's circles, some are triangles, some are squares, some are whatever. They all have these unique, shapes. And these particular receptors are going to be specific for that particular hormone. Do you guys see? So if I have a hormone that looks like this, sort of square, are you with me? Okay? So that would dissolve, not shown here, it would dissolve in the plasma, into the interstitial fluids, but it wouldn't bind to that receptor. Yes or no? No, because this does a round molecule. I just drew a square molecule. So the result is cyclic AMP is not elevated. Cyclic AMP is not elevated. It's going to have no effect. But let's say I now secrete this molecule, and that's the one that's depicted. Okay, it's a different hormone, just a completely different water-soluble hormone. Now, it does dissolve in the plasma. It does dissolve in the interstitial fluids. But now I have a receptor that will bind to that, you guys see? And then very quickly, it now begins to take ATP and make cyclic AMP. And the concentration of cyclic AMP goes up. As cyclic AMP goes up, what happens is it begins to catalyze a cascade of reactions. And so what happens, an enzyme is activated, then it activates another enzyme and another one. And as this cascade goes on, eventually what happens is whatever that cell is supposed to do, it does it at a greater rate. Whatever that cell does, it does it at a greater rate. Okay? And the result is that homeostasis would be restored. Does that make sense? Already? And so again, you have all these different hormones that are water soluble, but in all cases, the secondary messenger is the same. It's always the same. So what's the difference? Is the different receptors and the different hormones need to combine correctly. And then cyclic AMP goes up, and I'll have an altered cell function. Does that make sense? Okay, good. Alrighty. When you, the reason that this will be sort of important, and I've seen this, it's not a bad idea to get into the habit, particularly once you get through microbiology, and this course too. When you get a medication, but you guys know you get the medication, you get this piece of paper that's really long, you know what I'm talking about, or it's folded 100 times, and you can just unfold it and whatever. But when you read that, it will always tell you the mechanism by which the drug works. Okay, and it will rehearse this for you. Okay, it's in there. Okay, so far so good. So you would see this for sure. Okay, so the next one is going to be dealing with steroids in particular. Okay, ones that are water insoluble. And you guys remember from the male reproductive system, what we had those interstitial endocrinocytes, not the interstitial, but the sustentacular cells that secreted an androgen binding protein. You guys remember that? And so why do they do that? It was a protein. But the protein, of course, does dissolve in water. But my interstitial endocrinocytes would produce a steroid hormone called what? Testosterone. Now, testosterone does not dissolve in water. So what will happen is the androgen binding protein will bind to the testosterone, and now that will piggyback that hormone in the water-based system. Okay? And so that's what's happening here is I have a fat-soluble or a lipid-type hormone, and so there's always going to be some sort of a carrier molecule to sort of transport it in the water-based system. And so what happens, it does carry it through, and then eventually it comes in contact with my phospholipid bilayer. Now what I note, this is a steroid hormone, and here's my fatty acids. So would the steroid hormone dissolve in the fatty acid tails? Yes or no?
Yes, it would.
Yes, it would. And so it does. And so there it is. And so now it's inside that phospholipid bilayer. Now what I have to do is I have to get it inside the cell. So problem. Inside the cell, the cytoplasm is what based?
Water.
It's water-based. So again, what I need to do, once it's inside the cell membrane, now I need to get it out of the cell membrane and transport it into the cell. And so yet again, right here, see, I have another receptor. And so now that receptor picks up that hormone, and there it is. So now I have another, note, they're two different receptors, but they're carrying the same hormone. And the way this works is this. It can affect metabolic pathways, but it's going to do it in this manner. The receptor-steroid combination will enter into the nucleus, okay? And you guys remember you've got your DNA here that's divided up into these functional units called genes, okay? And each gene codes for a protein, which could be an enzyme or whatever, all right? And so what happens is the hormone receptor combo enters in and it stimulates the transcription of DNA into messenger RNA or an RNA transcript. Once I get my messenger RNA, it now goes into the cytoplasm, and there I have my whatosomes, ribosomes, and the ribosomes now do the process of translation. And what it's going to translate, of course, could be enzymes, okay? And the result is a metabolic pathway will go at a faster rate. Or it could be a structural protein, or it could be something else that's going on, but I'm now going to have an altered cell function. In other words, whatever that cell does, it's now going to do it at a greater rate. You guys follow? Okay. So those are the two biggies. right? Those are the two big mechanisms. You guys all set with that? All right. Hoi. Posterior pituitary gland. I want to show you guys something. Hold on. Whoops, hold on. I'm looking for a particular image which I know will be helpful to you. And I think this is it. This makes it real simple. Yeah, this is nice. If I can get it. Let me try one more time.
You can just copy and then put it into Word.
I don't want to do that. It's too open. Open a new window. Whoops. I lost it. Shoot. Can you guys see that okay? Nice. Okay, good. So what we're going to do is we're going to start talking a little bit more about the pituitary gland, which of course resides in the sella turcica, okay, of this sphenoid bone. And what's sort of important to note, everybody, is the pituitary gland is not one gland, it's two glands. It is two glands, and they have two totally different developmental origins, okay? And the result is they're completely separate glands. They're together, so sometimes we just like to just call it the pituitary gland. And what we do have here is we have a posterior lobe, and I have an anterior lobe. So when I'm dealing with this anterior lobe, everybody, that's going to be my hypothalamic hypophyseal portal system. And of course, what will happen is, and you might recall because we have talked about this before, but the hypothalamus is the control center. And then what it's going to do is going to secrete some releasing hormones or other hormones. We'll talk about that. And they'll go in through the fenestrations, through the hypothalamic hypophyseal portal vein, come in contact with the anterior lobe, and then it now is going to produce a particular hormone, and then off it goes. Do you guys see? But the posterior lobe is different, and what's going to happen here, you can see you have blood supply, but it's just your regular capillary bed. It'll be fenestrated. And but what I'm going to use and employ in this case, instead of releasing hormones, I'm going to be using neurosecretory cells. Okay, and so what will happen? The control centers are up here. The cell bodies of these neurosecretory cells will be in the control centers, and these neurons now, of course, will be part of my efferent pathway. And what they will do upon stimulation is secrete the hormone into the bloodstream, and then off it goes. Okay. So again, the only two hormones that the posterior lobe deals with is what? ADH and oxytocin. Those are the only two. And we're going to talk about those right now. A little bit. Maybe. All right? So, the first one here is the posterior lobe. And then I'm going to talk a little bit about the anterior lobe. That'll take a little bit more time. Okay, so I want you guys to grab your handout and turn to page 6. Can you guys see that okay? Because maybe we don't have to flip the lights on. You guys are all set with the way everything is? Good. All right, so we've dealt with ADH. And of course, you guys know that in the hypothalamus, we're going to have what receptors? Right, osmoreceptors. We're going to have what receptors, everybody?
Osmoreceptors.
You get that one right. Maybe? I think you probably did. And of course, what are those osmoreceptors doing? They are measuring the relative concentration of both salt and water. Okay? And so if water goes up, salt goes down, we're going to have a more hypotonic condition. But if salt goes up and water goes down, then I'm going to have a more what? Hypertonic condition. And when you have a hypertonic condition, what I want to do is I don't want to lose water. But if I have a hypotonic condition, I do want to lose water. And so what happens is this. If we have a hypotonic condition, if we have a hypertonic condition, what we want to do is we don't want to lose water. And so the result is the neurosecretory cells now are going to secrete antidiuretic hormone. Do you guys see? And so it goes into the bloodstream and then eventually goes to my kidneys. And of course, what the kidneys start doing in order to, to improve this situation and bring it back to the set point, the kidneys, the collecting duct and the distal convoluted tubule starts making what aquaporins? Aquaporins. Okay? And those aquaporins just start spontaneously embedding in that those collecting ducts. And now, as the urine, because at this point I can call it urine, as the urine begins to go through the collecting ducts, does it see the salty medulla?
With the aquaporins?
Yeah.
Yeah,
Yes, it does. And the result is it pulls in water by what? Osmosis. And at the same time, of course, as this is happening, my hypothalamus, when we talked about this, also stimulates what? Thirst. And so what we're going to do is we're going to go seek out and search for water, and we'll ingest that. Okay? And then, of course, it's going to have to, it's going to begin the process of restoring homeostasis. But again, I'm dealing now with water issues, and so it also makes sense that ADH not only is going to create the aquaporins to hold on to water, but it doesn't surprise us that from our sweat glands, it's going to cause them to sweat what? Less, so I lose less water. Okay? And then the other thing is this. You guys remember we talked about the urinary system and we looked at two types of regulation. We dealt with blood pressure and I dealt with water salt concentration. Do you guys remember? Okay, we looked at those two regulatory issues. The reality is the two of them are holding hands quite tightly. Do you guys follow? Because one will affect the other will affect back and forth. And so if you're having a blood pressure issue, you're probably having a water-salt balance issue as well. If you're having water-salt balance issue, you're probably having a blood pressure issue as well. And so all these systems sort of work synergistically. And so if I'm dealing with some water issues and there's not enough water, that also lets me know that perhaps my blood pressure is going down. And the result is ADH will cause your blood vessels to begin to constrict to help the process of raising blood pressure. Alrighty? And again, that'll be a negative feedback system. As I hold on to the water and I begin getting a more hypotonic condition, that'll have a negative effect, and the result is there'll be less ADH that's secreted. Okay? So it's all shown visually. So just you need to walk through that, and that'd be good. All right? Okay, next is oxytocin. I did that one already. I'm not going to do it again. Is that okay? So we did that on page 5. Now, posterior pituitary gland. Excuse me, anterior pituitary gland.